Yeah. So a very good evening to one and all. I think there are so many eminent speakers in this particular conference, and I have been listening right from the start. And there's so much to share, learn, and interact with all you people. Just before that, Ms. Diva, I think she has presented an excellent piece of work that she has done at such a young age. So my all the best wishes to you for a really rewarding career. So I'm Dr. Jyotirmoi Aich. I have done my PhD in asthma biology. Post that, I did my postdoctorate in cancer biology. And it's been 2-3 years that I've established a small research group. I'm basically working as an assistant professor in D.Y. Patil University. So where I teach a lot of students, and these are biotechnology students. So this is bachelor's and master's program as well. And simultaneously, since I have that, curiosity for the research, so I've established a small group. This is a very small study that I'm going to present, since I got an invitation, and it's such a nice platform to share some of the research that we do, and definitely to get inputs from the experts which are there in the panel. So it's definitely, I really thank the organizers for giving me this golden opportunity. So what I'm going to talk about, so I think drug repurposing, we have been talking, there have been so many topics already in this particular conference, and this is something which has really catch the news, or it has come really wonderfully in the field of therapeutics. So as Diva was also mentioning, that during the COVID period, okay, so when hydroxychloroquine, which was an anti-malarial drug, it was proposed to treat COVID, right? And then there have been so many other things like aspirin. So drug repurposing has really come out to in a new manner. So there are so many different areas that I'm working. Out of that, I have picked up one small work that I'm going to present, although the work is in preliminary stages. We are yet to confirm a lot of things, but still I wanted to share my whatever we have done, and in this particular conference. Okay, so the part that the topic of my presentation is old wine in a new bottle, drug repurposing of, just a second, I'm just trying to, drug repurposing of antipsychotic drugs. So now, so this we already know. Actually, this there's no more introduction needed, but still, just for the people who are a little unaware of the things. So, cancer incidence and mortality are rapidly increasing all over the world. In recent years, we have the biggest challenge that I think we have faced is the drug resistance. It's a chemo resistance problem that I think a lot of clinicians are facing since a lot of time. So, let's talk about some of the standard chemotherapeutic drugs like cisplatin, like doxorubicin. But again, there have been a lot of chemo resistance that the people that we are getting the case studies and everything. So again, there have been a lot of different ways also to tackle this chemo resistance. Drug repurposing could be one of the approach. So drug repurposing is one of the most widely used method that identifies new clinical medications for the existing drugs. So it is definitely going to be really helpful rather than going for an isolation of a new drug, right, which almost takes like 12 to 14 years kind of a thing. Drug repurposing can really cut short that particular time, but yes, it has got its own pros and cons. But if this can be utilized properly, it can definitely open a new era in the field of oncotherapeutics. So again, the figures are showing the distribution of different types of cancer, with the breast and the lung being the majority one, and the related mortality. What is the aim of this particular project? I don't want to go into the what exactly is a drug repurposing and everything, because everybody knows about it. I just wanted to show the piece of data that we have generated using using drug repurposing databases. So the main aim of the study is to identify a potential drug candidate that could be considered for drug repurposing in breast cancer, and to perform preliminary in vitro studies on the MCF. So we have taken in vitro model, which is MCF7, that's a breast cancer cell line. Objectives of the same, to identify a list of antipsychotic drugs. So we have chosen in this case an antipsychotic drugs because we have referred to a database which is called as a REDO, that is a drug repurposing database that has collated a lot of information based upon literature knowledge, based upon the the experiment studies and everything, and thereby it gives you a clue that okay, now these drugs can be taken further, but definitely there has to be a lot of in silico and in vitro methods, in vitro methodology that has to be put into before claiming something. So we identified a list of antipsychotic drugs that could have an anti-cancer potential. To identify the common genes involved in the breast cancer, so what we did is that we have identified some of the genes which are important in the breast cancer, and we have utilized the technology tool which is molecular docking again, which I think everybody is well aware. And I think with all these COVID time where we were not able to do a lot of experiments, I think this computation thing has really come up a lot, and it has really, it has really helped the molecular biologists to do a lot of different experiments. So we did a molecular docking just to identify from a pool of drugs available that which one could be having a much greater potential. Okay. Now molecular docking doesn't talk about that definitely, it is going to inhibit this gene or it is going to activate the gene, but yes, it can give us some clue how to proceed further. To select the best drug candidates based on the molecular docking studies and move forward with the in vitro studies, and to investigate the efficacy of the selected drug candidate by performing some in vitro functional assays. What is the basis now? The question might come that what is the basis of taking an antipsychotic drug? Now, when we have referred a lot of literature, so this is in the in vitro studies have shown that dopamine, okay, it is involved in the inhibition of cancer proliferation and migration. It was established in the year 2006, 2017. Recent studies have suggested that the dopamine D2 receptors are upregulated in the breast cancer. That is one of the major things, that these dopamine receptors are really upregulated in the breast cancer. So, and again, there's one more correlation. A majority of the antipsychotic drugs act as an antagonist of the dopamine receptors. Okay, so several studies have demonstrated that patients taking these antipsychotic drugs, they have low incidence of colon, rectal, prostate cancer when compared with the general population. Okay, thus an examination of the drug repurposing potential of these antipsychotic drugs in cancer would be appreciated. So we just summarized like what all, what are the known facts about this brexpiprazole. So we have taken one antipsychotic drug, which is a brexpiprazole, although we did a screening among the family of the antipsychotic drugs, and then we landed upon the brexpiprazole that it can be used. So this is an antipsychotic drug that is used in the treatment of schizophrenia. And this we have just collated from different papers that how brexpiprazole can be useful in the action in the different cancers. Okay, so that can be. So what is the research camp? The drug repurposing in a cancer has gained extensive focus, and currently the research on the drug repurposing potential of antipsychotics is extremely limited. The studies have further neither have performed in silico nor in vitro studies of brexpiprazole has been performed in the breast cancer. So what we did? So we referred, so from where we got this clue? We got this clue from REDO database, as I've already said. It is a drug, it is a repurposing drug in oncology, that is a REDO project. It has used a literature-based approach to identify licensed non-cancer drugs with published evidence of anti-cancer activity. So we have got a licensed non-cancer drugs again, which has a potential, or it has been shown to have a good anti-cancer activity. So this is a table from where we have taken the data, and brexpiprazole has been known to be better, but still we didn't want to just proceed with the brexpiprazole. We wanted to look from the pool of antipsychotic drugs. Okay, so we'll show you the next methodology. Yes, so this is the one. So we have identified the list of antipsychotic drugs based on the literature review and the REDO database. We identified the genes which are upregulated in cancer based on the literature review. We again selected the best drug candidate based on the results of molecular docking and the drug likeness study. And then later on, it has been followed by the MTT assay and Hoodley assay, and it is to be further supported by a lot of other assays also in order to confidently say that yes, this could have an anti-cancer potential. Okay, so this figure shows about that what are the different drugs that we have taken. We have taken a brexpiprazole, aripiprazole, olanzapine, serotinol, quinidipine, and the clozapine. And the different receptors. So basically the first tool that we've used is the molecular docking. So we did a thorough multi-docking analysis. We have taken some of the genes which are important in cancer, definitely. For example, overexpression of EGR has been related to so many different cancers, or there has been a deletion in the EGFR for exon 19, which is leading to cancer, lung cancer. So we have taken some of the hallmarks like EGFR, there's a survivin, we have this ER alpha, MAP kinase, HER2, PAC1, mTOR, and PAC4. Now, all these things, now see, when we talk about the genes which are important in cancer being a multi-gene disorder, it is just not possible to just list some of the genes, do your docking, and then you to go ahead again. So there could be like 100 of genes which are being important different types of cancer. We've just taken some hallmarks so that from a pool of the antipsychotic drugs we can choose at least one depending upon the interaction of these drugs with these ligands with the receptors, and then we can proceed further with our in vitro thing. So we have taken this. So from all these things, we can see that the more negative it is, the brexpiprazole, it is definitely the association is better. Further, we are also performing some MD simulations, some molecular dynamic simulation, in order to show how much stable is the interaction of brexpiprazole with EGFR, survivin, and the different receptors. So from this, we have taken the brexpiprazole to the in vitro studies. So these are just the molecular docking figures where we've taken the EGFR and the ER alpha. So the figure shows about like where exactly it is located and the interactions. So the number of when there is a more the number of hydrogen bonds which are represented by the green color, more stable is the interaction between the ligand and the receptor. So this is the again overview picture of your HER2. Then we have survivin, PAC1. These are the different receptors that we have taken. We've also taken mTOR, PAC4, and MAP kinase. And then again, we did a thorough analysis that where exactly which among all the antipsychotic drugs, which one is the most important one, right? So we also performed a drug likeness studies among the different drugs, and again identified the brexpiprazole to be a better candidate. Next, what we did is that once we have identified, okay, now brexpiprazole is considered, it could have an anti-cancer potential because it is binding nicely or it is binding more effectively with some of the receptors. Although those, computational data also has to be confirmed with different computation tools. But still, since this is just a 6 months project, the work that I'm showing. So again, we took a MCF7, which is an established in vitro breast cancer model. The cells were taken, the MCF7 cells were taken, and they were treated with different concentration brexpiprazole. And we identified the concentration, the IC50 value to be 10.14 micromolar. We also, there's the same thing again represented in a different manner. So when you see in the 10 micromolar, there is a 50% inhibition. So we can call this as an IC50 value. So these are the morphological alterations in the MCF7 cells treated with different concentration of brexpiprazole. Okay, so in treated cells looks fine. Your 5 micromolar, 7.5 micromolar also looks good. But the moment it has started with the 10 micromolar, the cells are shrinking as it is apoptosis has been initiated. So we are further in the process of doing a lot of checking for real-time PCR, checking for different apoptotic markers, doing a FACT staining. There are so many different things again, down the lane. We also performed a wound healing assay on MCF7, where we have shown that when the cells were treated with control, these are the control, and then two concentrations we have taken, this is a 5 micromolar and a 10 micromolar, we could see that the brexpiprazole is inhibiting the migration of the cells, which in fact it is better when you talk about the therapeutics in cancer. So these are just the preliminary activities. We did a quantification figures again for the wound healing assay. Later on, what we did is that now, since we got this clue that brexpiprazole is known to inhibit migration, so what we did is that we have identified some of the important, genes or some of the targets which are known to inhibit the metastasis process. Okay, so what we did is that, for example, it will be shown in the table later on. So what we did is that we have done a kind of a comparison between brexpiprazole and ropinirole. Now, this is ropinirole. It is known for I guess for the aromatase inhibitor, which is known to play an important role in the metastasis. Okay, so what we wanted to show is that the genes which are important in the metastasis, okay, now we have a known inhibitor against these genes. Okay, now if we do again a docking, and if the binding affinity is coming similar to that of the known drug, we at least can hypothesize that our drug also can prove better in inhibiting that gene again. Again, a lot of different studies are further needed. These are again, I will just take you down to the, just a second, yes, take you down to this particular figure, which is much more clear. Now, for example, there's the aromatase gene, which we know that it is known to play a very important role in the metastasis of cancer. Now, ropinirole is known to inhibit this particular gene. So what we did a kind of a comparison studies in order to see that where exactly this brexpiprazole is going to stand. And you can see the binding affinities are much better as compared to the standard. But yes, before proving that this definitely can also go for a much more potent inhibition, there are several more studies are required. So we have performed also for CCL18, NF-kappa B. There's a matrix metalloproteinases which are involved in MP9 and MP2, and there's a tyrosine kinase inhibitor. So again, we have this, for example, this SMC2158, which is a known inhibitor of the CCL18. Again, if you see in every time the brexpiprazole has come out, the binding energy has come down to be really better. So this has just given us a further clue, or it is a kind of a very strong preliminary evidence that we have got that brexpiprazole might have a very good anti-cancer potential, and then it can be treated, it can be used for the therapeutics in the case of a breast cancer. But yes, there's so many other studies which are also in the line where the students are working. So yes. The combined in silico and the in vitro approaches reveal that the brexpiprazole is a potential drug candidate for drug repurposing in breast cancer. The brexpiprazole, it has shown a stable and a significant binding interaction with various anti-cancer drugs. The in vitro cytotoxicity assay on the MCF7 cell also confirmed that brexpiprazole had a great anti-cancer potential. We did also a wound healing assay, which again revealed that brexpiprazole it has been shown to inhibit the cell migration. Metastasis analysis performed using comparative molecular docking suggested that brexpiprazole can play a role in inhibiting metastasis. So further, the migration assays and everything, we are in the process of doing it. Future prospects with more solid data from various functional assays, we hope to gain more evidence. Definitely, brexpiprazole can be studied in combination with the established anti-cancer drugs to study synergistic effects. So there's a combination model. I hope some of you must be knowing that, where you combine a chemotherapeutic drug with a repurposed drug, right? And then you put in combination. You just check out that which combination is going to be synergistic, which combination is going to be an antagonistic one. Okay, that kind of a thing. Then a quantitative real-time PSEC can also perform, which is again in the pipeline. As brexpiprazole has shown a multi-target potential, it can also be studied in different cell lines to study its effect in different cancer. Further in vivo studies need to be carried out to explore the cellular and the molecular mechanism of brexpiprazole in the tumor microenvironment. Okay, so these are the different kind of future prospects, and we are working on this. These are some key references.